It's now my great pleasure to introduce Matteo Trovato, who's a great friend and I would say even a better scientist. Matteo studied biology and specialized in molecular and cellular biology in Italy at the Scuola Normale Superiore of the University of Pisa in Italy. Then moved to EMBL in Heidelberg for his PhD in the lab of Dr. Minno, where he studied the role of H3 variants modifications on transcription regulation. He has recently defended his PhD, which was awarded cum laude, and is about to move to the IFOM in Milan to do a postdoc in the Pagani lab. So once again, Matteo, good luck, congratulations, and welcome to the Nucleosome session. We're looking forward to your talk.
Okay, thanks a lot, Matte, for the kind introduction. And let me start also by thanking all the organizers of the Fragile Nucleosome series. It's a fantastic space to share research, and I'm really honored to have the opportunity to tell you about today about some work that I did during my PhD. And in particular, I will tell you about the work that we did studying H3.3 and particularly the lysine 9 residue, and how we can use then also H3.3 not only to study this interesting histone variant, but also to use it in a way as a tool also to study more generally how histone residues and histone modifications function. And I'm sure for this audience, there is no need to introduce what histone modifications are. They are broadly characterized either as repressive or active, and they correlate fairly well with different transcriptional states. However, it is still a long-standing question in the chromatin biology field whether histone modifications really play a role in controlling gene expression, or if they are more of a consequence of the transcriptional state itself. And this is not a trivial question to address. And one approach that allows to do so is the histone mutagenesis, in which essentially what we do is to replace a modifiable histone residue, like a lysine residue, with a non-modifiable one, like, for example, an alanine. And by doing so, we expect that the downstream effects of this perturbation will be much more residue specific, for example, as compared to approaches that target the enzymes that are responsible for the deposition or the removal of the histone modifications. However, this approach is challenging to apply in mammalian systems in particular, because usually canonical histones are encoded by multiple genes. And even though there was recently the brilliant paper from the Halin lab, it is still quite challenging to introduce mutations in all these gene copies. And so for my PhD project, we decided to focus our attention on studying histone H3.3 variant, which is a histone H3 replication-independent variant. And from a technical perspective, studying H3.3 offers an advantage because this histone variant is encoded by only 2 genes, which makes, of course, much more feasible the introductions of mutation. But most importantly, H3.3 is also a very interesting variant to study by itself because contrarily to canonical histones, H3.3 is not uniformly distributed over the genome, but it is found enriched at very specific genomic locations, such as, for example, active enhancers or promoter of active genes, or also at specific heterochromatic regions. And H3.3 has always, over the years, and particularly in the beginning, has been always associated to active chromatin environments. And there are less studies that have focused on studying H3.3 in a repressive chromatin environment. And so for my PhD project, we decided to focus on the lysine 9 and lysine 27 residues of H3.3. And as a model system, I used mouse embryonic stem cells. And essentially, we started from a background mouse embryonic stem cell line in which one of the 2 H3.3 coding genes was knocked out. And then using CRISPR-Cas9, I introduced lysine to alanine mutation either at position 27 or at position 9 of the remaining gene. And then with this complete set of mutants, what we did was to generate several ChIP-seq datasets and also other genomic datasets, and also perform some differentiations and other phenotypic assays to probe the phenotype of these mouse embryonic stem cells. And so one of the first things that we did was to profile the transcriptional changes that occur upon introduction of these mutations. And as you can appreciate from the hierarchical clustering, both K9A and K27A mutants showed a distinct transcriptional profile from each other, but also distinct from the control mouse embryonic stem cells. And when looking a little bit more into the details of these gene expression changes, I usually like to represent them with this upset plot because we can see and appreciate to what extent the genes are, for example, shared between the two mutants in terms of upregulation, downregulation, and which genes are changing their expression individually in one or in the other mutant. And in the interest of time today, I will focus on the K9A mutant, and I think it is quite evident from the plot that we found this group of genes of over 1,000 genes that captured our attention because was selectively upregulated in the K9A mutant. And we're looking at what kind of genes were those. We found that they were mainly categorized in two groups according to the gene ontology enrichment analysis. So one group of genes was related to immune system processes, which is something that we typically don't expect to find expressed in mouse embryonic stem cells. And I will come back to this in the presentation. And then the other group of genes was instead related to development and differentiation. And these genes were also, to some extent, shared with the K27A mutant. And so altogether, with the K9A mouse embryonic stem cells, apart from the expression of developmental genes and immune genes, we also observed some specific features like an increased rate of cell death. And importantly, also, these mouse embryonic stem cells were unable to differentiate in vitro to any of the lineages that we tested, like, for example, the neuronal lineage or the mesoderm cardiomyocyte lineage. And so, of course, for us, it was then interesting to try to see whether we could identify changes at the chromatin level that could underlie these phenotypic effects that we observed. So one of the first things that I did was to profile the distribution of the repressive H3K9 trimethylation mark. And what we observed was that this histone modification was reduced at several genomic locations in the K9A mutant. And these regions with reduced K9 trimethylation signal were, not surprisingly, annotated as heterochromatic regions. And in terms of genomic distribution, they were mostly, like the decrease in K9 trimethylation was mostly occurring on distal intergenic regions. However, what was even more interesting for us to notice was that this decrease in K9 trimethylation was mirrored by an increase in K27 acetylation that was also occurring in the K9A mutant at regions that were annotated mostly as heterochromatic regions, and with a genomic distribution of these gained K27 acetylation peaks that was, again, kind of mirroring the ones of the lost K9 trimethylation peaks. So essentially, in the absence of the lysine 9 residue of H3.3, it seemed like we had some regions that were transitioning from a repressed chromatin state to a more active one. And to see whether these regions were indeed the same, we can visualize them as a heat map. And essentially, what I'm showing here are cluster 1 and cluster 2, the differential K9 trimethylation regions, with cluster 1 that showed a more dramatic reduction, and then this third group of regions is a group of regions with non-differential K9 trimethylation signal that I included as a control. And so when plotting the K27 acetylation signal side by side, we can appreciate that this gain in K27 acetylation was indeed occurring at the same regions that were losing K9 trimethylation. And there are a couple of interesting observations regarding these genomic regions. And the first one is that when we profiled the distribution of H3.3, we found that cluster 1 and cluster 2 regions were showing enrichment for H3.3 as compared, for example, to the non-differential K9 trimethylation regions. And also importantly, the deposition of H3.3 was not reduced in the K9A mutant, indicating that this loss of K9 trimethylation that we observed was indeed a consequence of the removal of the residue. And another interesting observation for me was to see that this cluster 1 group of regions was showing already kind of a baseline K27 acetylation signal already in the basal condition, despite the presence of the repressive K9 trimethylation mark. And then upon removal of the K9 tri mark in the K9A mutant, we see that these regions gain quite an abundant K27 acetylation signal. So this kind of suggested to us that maybe these regions are already somehow primed to be activated already in the basal condition. And to verify this hypothesis, what we did was to integrate our ChIP-seq analysis with publicly available ChIP-seq data that are obtained from the ChIP-Atlas database. That is essentially a collection of ChIP-seq from chromatin factors and transcription factors from wild-type mouse embryonic stem cells. And when checking the enrichment from all this data, we found that indeed cluster 1 regions were showing a relative enrichment for several transcriptional activators and co-activators, like, for example, P300, bromodomain containing protein, or even pluripotency transcription factors. While on the other hand, cluster 2 regions, so the ones that didn't show this baseline K27 acetylation level, were showing enrichment for more canonical heterochromatin binders. So in the K9A mutant, we now have some regions that gain K27 acetylation on distal genomic regions. And usually when we have this mark at distal genomic regions, this typically speaks for putative enhancer elements. And usually when enhancer elements or cis-regulatory elements are in an active state, it is possible to measure an increase in the levels of nascent transcription, which are also called as enhancer RNAs. So I did a PRO-seq experiment to measure indeed the nascent transcription signal, and we found that particularly cluster 1 regions showed an increase in the levels of nascent transcriptions in the K9A mutant in particular. Again, to suggesting us that these regions could potentially function as regulatory elements. So the consecutive step then was to try to see whether we could identify putative target genes of these regulatory elements. And for this, former postdoc in the lab, Daria, constructed a gene regulatory network using a tool that was recently developed in the Judith Zaug lab, also from here, EMBL Heidelberg. And by doing so, we could identify genes that were linked to cluster 1 and cluster 2. And when looking at their expression, we found that these genes were selectively upregulated in the K9A mutant. So of course, then we wanted to see whether this increase in the expression of genes selectively in the K9A mutant was essentially linked to the ones that I showed you in the beginning of the presentation, just from the RNA-seq, and whether we could identify again this group of genes. And so one of the first things that I did then was to see, since we expected genes related to development and differentiation, I inspected the proportion of genes linked to cluster 1 and cluster 2 that were showing a bivalent promoter signature. And this is because, as many of you know for sure, genes with a bivalent promoter in mouse embryonic stem cells are usually related to differentiation, and then they are the bivalency of their promoter, so the presence of activating a repressive histone mark, is typically resolved throughout differentiation. And so we found that about 20% of the genes linked to cluster 1 and cluster 2 were showing indeed a bivalent promoter signature. And this is an example that I'm showing here, in which we can see that a cluster 2 region in which the K9 trimethylation signal was lost in the K9A mutant showed also a concomitant increase in K27 acetylation, and then through the GRN, we identified the TBX20 transcription factor as a putative target of this region that was showing increased expression in the K9A mutant selectively. And I don't have the time to go into the details of this, but if you notice here, we also observed a reduction in the K27 trimethylation on the promoter of this gene. And we have some data that were also generated by a collaborator from the Davidovich lab, that showed that potentially the K9A mutation might also, or the lysine 9 residue of H3.3 might also be relevant for establishing the K27 trimethylation on the promoter of these genes. But altogether, in the interest of time, essentially, we found that the upregulation of these genes could be indeed related to the activation of these distal regulatory elements and the fluctuation in the repressive histone modifications. But what about the other group of genes related to immune system processes? Was this also the case for these genes? And actually, so when we performed gene ontology enrichment analysis for the genes linked to cluster 1 and cluster 2, we found again a significant enrichment for terms that were related to immune processes. And I would like to also highlight here that these genes were not just related to general stress response, but the underlying genes, so the genes underlying these terms, are also genes that are typically expressed in very specialized immune cell types, like, for example, MHC class 2 genes or toll-like receptor genes. And this is, again, one example in which you can see a cluster 1 region that shows decreased K9 intramethylation in the K9 mutant and increased K27 acetylation, and then the gene that was linked to this region through the GRN, the TLR2 gene, is showing increased expression in the K9 mutant. So we did several analyses to verify that indeed this upregulation of immune genes was somehow or could be somehow driven by these distal regulatory elements. And I will show you just one of these analyses that we did, in which essentially what I did was to take enhancers that are typically active in immune cells, which were retrieved from the mouse cis-regulatory atlas, and then we checked the overlap with our cluster 1 and cluster 2 regions. And when comparing to the non-differential K9 intramethylation regions, we found significant overlap between active enhancers in immune cell types and our cluster 1 and cluster 2 regions that were losing K9 intramethylation in the mutant. And also when looking at the activity of these enhancers in the different immune cell types, again, using the data from the mouse cis-regulatory atlas, we found that apart from stromal cells, these enhancers are typically active in quite specialized immune cell types. But to conclude then, from a chromatin biology perspective, of course, we wanted to try to understand why the lysine 9 residue of H3.3 is so important to maintain such specific genomic regions in a repressive state. And some of you might know already where I'm going with this, but of course we turned our attention at transposable elements. And this is because transposable elements in mouse embryonic stem cells are maintained in a repressed state by K9 intramethylation. And then it is also known nowadays that transposable elements represent an important reservoir of sequences with a regulatory potential. And among transposable elements, we particularly focused on endogenous retroviruses or ERVs, because H3.3 was suggested before to be important to maintain ERVs in a repressed state in mouse embryonic stem cells, even though there has been debate over the years on the function of H3.3 in this mechanism. And also a very important bit of information in this is that also ERVs have been co-opted throughout evolution to function as enhancers in immune cell types. So taking all this knowledge into account, it made sense to us that this ectopic upregulation of immune genes in mouse embryonic stem cells could be due to the repression of ERVs and then the activation of like cryptic regulatory elements that derive from them. So what I did was to perform a transcriptional analysis of ERVs, and we found indeed that ERVs were selectively upregulated in the K9A mutant. And also when looking at the ChIP6 signal on ERV sequences, we found again that the K9 intramethylation signal was affected specifically and with a concomitant increase in the K27 acetylation signal. And also to kind of close the circle, then what we did was to check the presence of ERV sequences within cluster 1 and cluster 2 regions. And again, we found a significant overlap at these regions compared to the non-differential K9 intramethylation regions. And these are the two examples that I showed you before, development-related gene and an immune gene, in which the cluster 1 and cluster 2 regions encompass ERV sequences. So this brings me to the end of the presentation. And by studying the K9A mutant, we found that these cells show a very peculiar phenotype already at the stem cell state. They fail to differentiate, and particularly they show upregulation of very specific group of genes, like genes related to differentiation and immune genes. And again, from a chromatin biology perspective, my interpretation of the data is that by studying these two mutants and the comparison between the two, we can see that the reduction in the repressive histone modifications can be then linked directly to the activation of the genes that we observe. And to put it as a schematic, so I didn't have the time to show this to you, but when we mutate the lysine 27 residue of H3.3, we see that this is enough to decrease the K27 trimethylation level at the promoter of several genes, and this is sufficient to trigger the activation of quite a few of them. But for many genes, this is not enough to trigger their activation, and despite the reduction in K27 tri, they remain in an off state. And this is where the comparison between the K27A and K9A mutant comes particularly useful, because we actually see that many genes are kind of maintained in a repressive state by a dual repressive mechanism. And it is actually the K9 trimethylation on the distal elements that is the prominent repressive mark. And then when we lose this mark in the K9A mutant, we trigger the activation of cryptic regulatory elements that potentially drive the expression of these genes. Also, to conclude, as it was recently also presented in a preprint from the Banaszewski lab, we have data showing that probably the activation of these cryptic regulatory elements is also triggering a rerouting of transcription factors, and we find that canonical enhancers in stem cells are affected in their activity, leading to a general rewiring of the gene regulatory networks in the K9A stem cells. And so with this, I would like to conclude by thanking my supervisor, Min, of course, for the supervision, for the constant discussions and the work that we have been doing over these years. And also Daria, Umut, Nadine, and Anna that were primarily involved in this project, all the current and past members of the lab, my thesis advisory committee, the Embryonic core facility, the collaborators from the Davidovich and Garcia lab. And if you're curious to know more about the work that we did, you are more than welcome to check the preprint on bioRxiv. And if you have any questions or comments, I would be more than happy to discuss with you. Thanks.
Fantastic talk, Matteo. Thanks a lot. That was very smooth, rounded up. So maybe I can start with a question that is always close to my heart while I already see that whether are starting to pop in. But regarding the position of K27 tri in the K9A mutants, you know that PRC2 comes in several flavors. I was wondering whether you checked whether you have the differential enrichment of either PRC 2.1 or PRC 2.2 on those regions, and if you know which part, if any, specifically is used in this case.
Yeah, so we didn't do this experiment, because in the end, exactly the story focused more on these distal elements. But essentially, what we found is that Polycomb seems to be still recruited at the promoter regions of the developmental genes that show increased expression in the K9A mutant, and this again is nice in the comparison with the K27A mutant, in which we see actually that the PRC2 complex, which we profiled by SUSY12 chip, is more dramatically reduced. So we don't know exactly what is happening at the in terms of Polycomb and with the K9A mutant, but taking together the genomics experiment and the biochemical experiments that were done in the Davidovich lab, it seems somehow that the lysine 9 residue, it might affect the activity of the PRC2 complex, because we see the decrease in the mark also in the biochemical experiments, but we didn't profile differences in the PRC2 one versus
Okay, great, thanks. We have a question from Pradeep in the box. Is that did you not see any upregulation of line 1 elements if you look at them?
No. So we didn't see in one of the supplementary figures of the preprint, we have a more detailed analysis of the transposable elements, and we only see enrichment of H3.3 at ERVs, and at some sign elements. So line elements don't show much H3.3, at least from our data. So it makes sense then that the mutation doesn't affect the expression of those elements.
Okay, thank you very much. I think we'll need to wrap it here because we are running short of time, but there's another question in the box if you'd like to answer that, and seeing that Carmelo's the hands up, so if you would like to type it, Carmelo, as well, and then maybe Matteo can answer it in written in the Q&A box.
Yeah,
thanks again for a great talk, and I'll
Thank
pass
you.
it over to Sky for the next talk.
Yeah, thank you.